Hopefully you can see it. And this was an interesting one. It was done in the so-called Welsher lab at Duke University. So, as people know, I went to Duke. So that means it must be true. And this was a published study that I don't know, it was the first time where they actually were able to find a virus and track it as it infected the surface of human intestinal cells. So this was a landmark study in the real-time uh, um, documentation, let's say, of a virus infecting human intestinal cells. So let's take a look. Okay, so there you saw it. Now, some of you of the more skeptical bent might actually wonder, how did they know that that purple squiggly line was actually a virus? So I decided that even though, of course, if it was from Duke, it must be true, I was going to go look at that study and go and find it and see how they knew it was a virus. So let me take a look at that. This one, we don't need to share the sound, but we're going to share this, share the screen. And here's the study. And as usual, it comes in this wonky format, which it's actually the study itself had a lot of pictures, but the pictures don't come out in the way that I do it. But anyways, you can find the study if you want. It was from a journal called Natural Methods, I guess, 2022, November 10th. There's the pages capturing the start point of the virus cell interaction with high-speed three-dimensional single virus tracking. So they're claiming that this purple squiggly line was a virus that they watched infect and attack the cells. So they say in the abstract, the early stages of virus-cell interaction have evaded observation due to the rapid diffusion of virions in the extracellular space and the large three-dimensional cellular structures involved. Here we present an active feedback single particle tracking method with simultaneous volumetric imaging of live cell environment to address this knowledge gap, to present unprecedented detail to the extracellular phase of the infectious cycle. So then you go to and it goes on, obviously, and all of this stuff is basically explaining how they get these images. So I'm not going to go through that because frankly, I didn't read it all. But it was all about this 3D smart image, and the 3D faster imaging system collects sequential volumes, et cetera, and they get all these pictures. And the first thing I noticed was there was no methods section in the body of the paper, which is weird. So they go right from the introduction to the results, and then they talk about how they essentially it's the same getting into how they got these images and how they labeled the virus, and the different kinds of viruses that they did, and the microscope and the pixel dwell time of the raster scan, whatever that means. preventing the blur motion or the motion blur, and all this stuff, which is mostly about how they got the images. Um, so then they tell you that these were the surfaces. And again, those of you of a more skeptical note will say, yeah, but how did they know that was a virus? Where did they prove that that was a virus? So again, it goes on and it's pages and pages and then discussion, internalized viral tracking. And then finally, in the sort of after part of the paper, we get to the methods. And again, it was all about this 3D smart thing and the instrument and how they got the optics and the overview of the faster thing and the excitation optics, imaging, variable, focus, detection. All this is basically optics, shared emission pathway. And you say, I just want to know where they got the virus from. So then they track an imaging integration, trim data acquisition flow work, automated acquisition, trim data, let plasmid construction, uh, production of this. And finally, almost at the end, we finally get to viral vectors. That means the viruses were produced by the Duke Viral Vector Core Facility as described previously. So briefly, they took these HEK 293T cells grown in 10 centimeter plates, were transfected with this calcium phosphate based protocol, and they grew the viruses in the cell culture. And then they took some of the materials of the cell culture and quantified how much of this there was. Then the viral solution was stored in aliquots at minus 80 degrees. And then they give you the details of the cell culture grown in these immortalized HeLa cells at the Duke Cell Culture Facility, and et cetera. So, and, supplemented the same old thing grown using the restricted media, supplemented with fetal bovine serum, using penicillin and streptomycin, and maintained at 37 degrees. And then you got something that they say was the growth of the virus, and then they stained it, and then they dumped that in and did all the optics and got the pictures. So, as I keep saying, and hopefully everybody has heard this so they're sick of it by now, virology is all about the so-called viral culture, otherwise known as the cell culture. If the cell culture is bogus, then they're not dealing with a virus, they're dealing with a broken down cell culture. And I think all of you know that the cell culture, starting with the 1954 paper from Enders and many, many times in the medical literature, has been shown that you get the same results whether you put a sample of anybody in a cell culture or whether there's no sample at all. You still get the cytopathic effect, proving without a shadow of a doubt that there is no virus. The virus is never the independent variable in a cell culture. They never do proper controls. We have proven over and over again that there is no possibility of showing the existence of a virus with the way they do cell cultures. So essentially, once again, all they were doing was dumping the cellular debris, breakdown products, staining whatever stains, dumping that in somebody's intestinal cells culture, taking these fancy images and claiming that somehow shows viruses entering the cell or the intestines without ever once showing that they actually were working with the virus. So, as they say, it's the same old story everywhere you go. You get scammed and libeled, and you hear words you never heard in the Bible because it doesn't say anything about this kind of nonsense there. So that's the story with that. So unfortunately, we have to put Duke, which I already knew, in the same boat with everybody else as doing pseudoscientific, fraudulent research.